All right, so good morning, everyone. So it's a pleasure today to welcome Aline Muyle for this same seminar. So we'll just briefly introduce her in case you don't know her. And so Aline obtained her PhD in 2015 in Lyon. She was working with Gabriel Marais, and she did her PhD on sex chromosome in Silene. And she's been discovering a very interesting early dosage compensation on those young sex chromosomes. Then she stayed for a while for 2 years in her lab, in a PhD lab, and which was also very productive, as she discovered some very interesting imprinted-based mechanisms of dosage compensation in those sex chromosomes. Then she finally got to a postdoc abroad. She went to California in Irvine in the lab of Brandon Goat. It was thanks to EMBO and Human Frontier fellowships. And there she's been working on related topics in epigenetics. That's the topic of our talk today. So Aline has been, she was hired as a CNRS researcher. She just started like a few weeks ago back to Lyon, in LBBE. And so it was very recent. She's coming back from California, and she will be presenting a work that she did there. So she obtained many prizes. She did many great papers, and so we are really happy to hear what she's been doing there. And so Aline, thanks for accepting to give this video seminar, and over to you.
Thank you very much, Thomas, for the kind presentation. It's a pleasure to be here, and thank you all for joining from Montpellier and elsewhere. I'll start sharing my screen, and I hope you can see it okay. Yes. So I'm going to present 2 projects today from my postdoc with Brandon Goat in California. And both projects are related to epigenetics. So epigenetics has many definitions, but my favorite one is the one that you can see here. And it is the study of changes in gene expression that can be inherited through cell divisions, either mitosis or meiosis, and that are not due to changes in DNA sequence. So with this definition, epigenetic marks can be either DNA methylation, histone modifications, or small RNAs, and even other things. And it is still a question in the field whether epigenetics can play a direct role in adaptation. And like Cavalli and Hurd said, it would be an important conceptual change in evolutionary biology if we could demonstrate that other molecules than DNA can carry substantial heritable information. And up to now, it has been unknown whether epigenetics can be important for adaptation, because it is actually quite difficult to disentangle epigenetics from genetics. And the requirements for epigenetics to be under selection and to play a role in adaptation is that it must affect fitness, obviously. It must also be inherited through generations and epimutate over time. So during my talk, I will focus on DNA methylation, which is an epigenetic mark. And I'll start with a brief introduction, and then I'll follow with the presentation of my work. So DNA methylation, as you may know, is a modification of cytosines by adding a methyl group to the carbon number 5. And in plants, it can happen in different contexts. So there are 3 contexts, either CG, CHG, or CHH. And H can be any base except for G. And we will see that the context matters because the different contexts play different roles. And CG context is symmetrical. Both strands, DNA strands, can be methylated. The CHG is also symmetrical, but the CHH context is not. And one method that can be used to characterize DNA methylation is bisulfite sequencing, also called BS-seq. And in this method, you start with the DNA, which is methylated or not at cytosines. And both strands are then treated by bisulfite, which converts non-methylated cytosines into uracil, and the methylated cytosines are protected. After this treatment, there is a PCR amplification and Illumina sequencing. And when the reads are mapped onto a genome reference, you can tell what cytosine was methylated or not. And you can do that in a strand-specific manner. So after this DNA methylation sequencing became more available, people got interested in looking at how DNA methylation is transmitted over time. So for that, they used epimutation accumulation lines in Arabidopsis thaliana. Starting from a parent, they studied it over many generations of selfing, and they sequenced the progeny after about 30 generations. And looking at the methylation divergence over generations in the CG context, they observed that the divergence increases over time. And so this is the evidence that CG methylation is inherited over generations, and it slowly epimutates over time. And they estimated the epimutation rate to be of the order of 10⁻⁴. So this is a lot faster than genetic mutation rate, which in Arabidopsis is around 7 10⁻⁹. And so this is important because it shows that epigenetic information it can be uncoupled from genetic variation. And so the fact that CG methylation can be transmitted and epimutates over time is necessary for it to be under selection. The observation is quite different in the two other contexts. In CHG and CHH context, methylation divergence is constant over time, over generations. And so it suggests that for this context, the methylation level is determined by intrinsic factors, such as genetic factors. So all this to say that CG methylation is the best candidate to be under selection and play a role in adaptation. So let's look now at the patterns of DNA methylation in Arabidopsis thaliana. And this is in genes. So in this figure, all genes in the genome were combined. And you can see here the methylation level inside the gene and upstream and downstream in the three different contexts. And genes in Arabidopsis thaliana tend to be CG methylated only with a depletion at the transcription start and end. And they are not methylated in CHG nor CHH in general. And this is quite different from transposable elements, which are usually methylated in the three contexts, and this silences them. So this was at the all genes combined, but when we look gene by gene, we can infer the state of the gene when comparing it to the average across all genes. And so some genes can be categorized as unmethylated. Other genes have only CG methylation, here represented by the black lollipops. And these genes are called gene body methylated genes. Other genes are methylated in either CG or CHG context. And finally, some genes are methylated in the three contexts. And people have found that these different methylation levels are associated with different expression levels. For example, the CHH methylated genes are not expressed, they are silenced, like transposable elements. And the gene body methylated genes are the ones that tend to be the most highly expressed. Here in this figure, people looked at the methylation state of orthologs across species. So here you have various plant species with their phylogeny on the left. And each column here is one ortholog across the species. The white color is for unmethylated genes and blue for gene body methylated genes. I will use on my slides the GBM abbreviation for gene body methylated from now on. And a pattern that you can clearly see on this figure is that the same orthologs tend to be gene body methylated across plant species. And so this suggests that gene body methylation might be important because it is conserved across species. It is, however, a very variable trait. So here you have on the right the percentage of gene body methylated genes in each species. And you can see that this varies from 0 to up to 60%. And so some species completely lack gene body methylation and others have it very much. So we can wonder what determines this diversity of gene body methylation in plants, and does gene body methylation matter for fitness? And this has been a very controversial topic over the past 15 years since gene body methylation was discovered. And I'm going to list here the arguments that are in favor of an important role of gene body methylation for fitness. So one argument is that gene body methylated genes tend to be intermediately to highly expressed, and they have a high expression breadth across many tissues. They evolve more slowly than other genes, and they are conserved across species. Here you have two grass species with their gene methylation level, and you can see it's highly correlated. And this happens even over 300 million years across plants, which suggests it is important. Other arguments have been used to say that gene body methylation is just a byproduct of something else and that it doesn't matter. People have said that because it has been lost in different species, it is not an important phenomenon. However, we know that it happens very often in evolution, that some characteristics are lost, like even mitochondria have been lost in some eukaryotes. So this shouldn't be used as an argument, I believe. Another argument that was used to say that gene body methylation is not an important phenomenon is that people have said that when it is lost, gene body methylation, it doesn't affect gene expression. So this was a study by Bewick et al. In 2016. And when I reused their data to study that question with my PI, Brandon Gart, we found the exact opposite answer. So we found that the loss of gene body methylation affects expression. And so we published it. And then the first authors of the first study, they answered again. So this is a very controversial topic. And today I'm going to speak more about gene body methylation and whether it's an important phenomenon or not. So I'm going to present a work that we recently published in Genetics. And this was a collaboration with Jeffrey Rossi-Barra and Daniel Seymour. And of course, my PI postdoc supervisor, Brandon Gart. So the hypothesis we wanted to test is that gene body methylation is important. And so if it is, then variants that lose gene body methylation should be removed from wild populations. And interestingly, we can use the site frequency spectrum, or SFS, to estimate the efficacy of selection acting on gene body methylation at the gene level. So we used the great data set available in Arabidopsis thaliana of over 1,000 methylomes, and the plants were sampled in the wild across the world. Here you can see the map of the samples in Europe. So we used only leaf methylomes, around 900 samples that we down sampled to 600. And for each gene in each accession, we looked at whether the gene was unmethylated, which is abbreviated or gene body methylated. So methylated in the CG context only. And using that information, we can plot the site frequency spectrum. So here you have the number of genes on the y-axis, and on the x-axis, you have the number of unmethylated accession. So when there is no unmethylated accession, it means that the gene is fixed for the GBM state, the gene body methylated state in the population. All individuals are gene body methylated. And on the contrary, when there are 600 unmethylated accessions over 600, it means that these genes are fixed for the unmethylated state. In the entire population has the unmethylated state for these genes. So this is our site frequency spectrum for all genes taken together. And we also used two outgroups, Arabidopsis lyrata and Capsella rubella, to infer the ancestral methylation state of each gene by parsimony, because we thought that the genes that are ancestrally gene body methylated might have different selective pressures. So here you can see the SFS for all genes that I just showed before. And here the SFS for ancestrally gene body methylated genes. And you can see it's a completely different SFS, which is skewed to the left for the fixed gene body methylated state. And here the SFS for ancestrally unmethylated genes is skewed to the right for the unmethylated state. And so the question we want to answer is whether methylation is under selection in Arabidopsis thaliana for genes. And so to answer that question, we used a model developed by Charlesworth and Jane. So it is a classical population genetic model, except that it was slightly modified to tackle epigenetics and its higher epimutation rate compared to genetics. So the model starts with two states, gene body methylated and unmethylated, and each state can epimutate to the other with epimutation rate mu and nu. And the shape of the expected site frequency spectrum is determined by the following parameters: the effective epimutation rates and the effective selective coefficients. So we use S unmethylated for the selective advantage of the unmethylated state, and S GBM for the selective advantage of the gene body methylated state. And we estimated all these parameters using an MCMC random walk. And once we inferred the value of 4 NES, the effective the selective coefficient, then we infer that if it is higher than 1, then there is significant selection on the methylation state. So back to our data, here we have the SFS for all genes with the observed and expected SFS. And this is for about 22,000 genes. And here is the value of the 4NES parameter that we estimated for NES for unmethylated. You can see the parameter is very close to 0. And for the GBM, the gene body methylated state, it was even closer to 0. So we can conclude that it is lower than 1. So there is no selection on gene body methylation when looking at all genes together in Arabidopsis thaliana. But now looking at the genes that ancestrally are gene body methylated in this group of plants, And this is for about 3,000 genes. And you can see for any SGBM, here the selective advantage for the gene body methylated state is significantly higher than 1. And so we can conclude that for these genes, the gene body methylated state is advantageous, which is probably why these genes remain gene body methylated over time, because there is selection to maintain that state. And then looking at ancestrally unmethylated genes, we can see that the selective coefficient for the unmethylated state is significantly higher than 1. So we can conclude that the unmethylated state is advantageous for this gene category. So we can see that there are different different directions for selection depending on the gene. And we did various controls to verify these results. We categorized genes into the ones that have different epimutation rates to check that there was no bias. We also redid this analysis at the level of cytosines, because here the results that you see are at the level of gene, and we could confirm the results. So we can say that there is selection acting on the methylation state of genes in Arabidopsis thaliana. Then we wanted to know, so why is this gene body methylation under selection? So we looked at the expression level of genes. Here you have on the y-axis the normalized expression level, and on the x-axis you have a random sample of genes in Arabidopsis thaliana. And for each gene, in pink, you have the expression level for the accessions that have the gene body methylated state, and in light blue, the accessions that are unmethylated. And so you can compare visually, for example, some genes have a higher expression for the gene body methylated state, and sometimes it's the opposite. But when we look at all genes across the genome, there are 52% of genes that are more highly expressed when they are in the gene body methylated state. And this is significantly more than expected by chance. So we can say that the gene body methylated state is associated with a higher expression level. And we also detected it with a linear model. So it is a small effect, but it is significant. And we also detected that the gene body methylated state has a lower coefficient of variation. You can see it here. It often happens that the individuals that have a gene body methylated state have a much less variable expression level compared to the unmethylated state. So gene body methylation is associated with a higher expression and also a more stable expression level. So to conclude this first part, we saw that gene body methylation is under significant selection, suggesting that it has a function, and so it must affect fitness. The selection coefficients that we estimated are quite small, of the order of 1.4, and this is actually similar to the magnitude of selection acting on codon usage. We could observe that gene body methylation is associated with a slightly but significantly higher gene expression. And we observed that in two different studies. And interestingly, there is another group working on that question, Daniel Zibelman, and they recently put a manuscript on bioRxiv where they observed that gene body methylation variation is associated with drought tolerance and flowering time in Arabidopsis thaliana. And so this suggests once again that gene body methylation is important for important traits. And so it is something that can be under selection in natural populations. For example, if a gene impacts flowering time, and if when it is more highly expressed, flowering time is delayed, and delayed flowering time is advantageous, then selection can act on the gene body methylated state. And this state is transmitted to the progeny, so it can increase in the population in frequency, just like a normal polymorphism would do under selection. So it is an important finding, at least in plants, that epigenetics can play a role in adaptation. So until now, I have mostly spoken about gene body methylation, which is only in the CG context in genes. But in plants, genes sometimes are methylated in the 3 contexts. And so I'm going to focus on this in the second part of this presentation. And the methylation in the 3 contexts in plants is regulated by RDDM for RNA-directed DNA methylation, and it is controlled by small RNAs. And this phenomenon is mainly directed at transposable elements, TEs. And we know from quite a few years now that transposable elements can affect gene expression when they are located nearby. So you can see that on this graph. Here you have the expression of the gene depending on the distance from the closest transposable element. And you can see that genes that have close by transposable elements are less expressed than other genes. And so this has been used to infer that methylation of the transposable element can spread in cis to nearby genes and prevent the proper expression. So a transposable element can affect gene expression in cis. And a question that is still open in the field is whether transposable elements can affect gene expression in trans, so when they are located far away from the gene. And this is the question that we asked in this second project that I'm going to present, that we published recently in Molecular Plants. So this was a collaboration with Alex Boussios, who works in Brighton in UK. So one way by which transposable elements could affect gene expression in trans is through gene capture. So here on this figure, I represent a normal transposable element, which we call free, and here a normal gene, which we call free also. And it sometimes happens that one transposable element can capture a fragment of a gene, here represented in green, and this leads to homology between the transposable element and the gene. And this homology in sequence means that small RNA will be able to map both to the transposable element and to the gene itself. And Lish has hypothesized that this could be used by the transposable elements to hide themselves from the host response. So the host usually tries to methylate transposable elements so that they are silenced and they cannot multiply in the genome. However, if the host here silences the element by methylating it with small RNAs, these small RNAs can also map onto the gene, which was captured, and methylate the gene, which would be disadvantageous for the host. And so there is a conflict arising from this phenomenon that could benefit the transposable element. So previous studies have looked at this gene capture in rice, but they didn't find much evidence of epigenetic conflict. So we wanted to look more into this question with this project. So here are a few predictions that we made. First, the genes that are captured, we call them donor genes. And we expect that they should have increased mapping of small RNAs and increased methylation level compared to genes that were not captured. So this is one prediction that we made. Another prediction is that maybe if this gene capture happens to a gene which is very important for the host, then maybe selection will limit these trans epigenetic effects for these important genes. Another prediction that we do is that these small RNA mapping and DNA methylation for important genes, they should not spread beyond the captured region. So they should be just located on the sequence that is homologous to the captured fragment for important genes. And finally, we made the prediction that maybe the gene capture by the transposable element might benefit the transposable element by evading the host response. So let's start by the first prediction. First, I'll explain how we annotated the gene capture events. So we filtered genes in the maize genome to have a very clean set of genes that we blasted onto a very clean set of transposable elements. So we used different types of transposable elements, rolling circle, DNA transposons, and also LTR retrotransposons. And so by blasting the genes onto the transposable elements, we were able to identify 6,000 free transposable elements and 1,200 that have captured a gene fragment. Often it happens that transposable element captured different genes with different fragments. And we have 23,000 free genes and 1,600 donor genes that were captured. Here we look at the level of small RNA per kb mapping onto the exons of genes that were captured, the donor genes, compared to free genes. And we can see that donor genes map significantly more small interfering RNA, which is what we expected. So it shows that there is mapping of small RNA on both the TE and the gene that was captured. And then we can wonder if this small RNA mapping leads to methylation of the gene. And so we look here at methylation level in CG, CHG, and CHH context. Here you have the proportion of genes on the y-axis, and on the x-axis, different bins of methylation levels from 0 to 100%. And so what you can see here in dark blue, the genes that have been captured are much more highly methylated than the genes that were not captured, which shows that the genes that were captured are indeed more highly methylated due to this transposable element capture. So we can confirm the first hypothesis that we made. And now onto the next hypothesis, which is that maybe selection limits these effects for important genes. So to look at important genes, we used something that is known in the literature, in the maize literature, is that synthologs are usually have important functions compared to translocated genes. So translocated genes are genes that have moved. Here we annotated them, either they moved in the maize genome or in sorghum genome, we cannot tell. But so we looked at the order of genes, and when only one gene has moved, then we call it translocated. And if it stayed in the same order in maize and sorghum, we call it syntholog, and we put it in the category of important genes. And interestingly, we saw that the proportion of genes that are captured by transposable elements differs between synthelogs and translocated genes. So here you have 3 genes. So the genes that were not captured by transposable elements, they contain 11% of translocated genes. And this is significantly different from genes that were captured, donor genes, which tend to be much more often translocated. So this suggests that maybe the capture by the transposable element triggers gene movement in the genome. So now that we have this category of important genes, synthelogs, and less important genes, translocated genes, we can separate them in our analysis to see if they are different. So here we have small RNA mapping per kb on the y-axis in donor synthelogs and free synthelogs, so genes that are important. And you can see again that genes that were captured map more small interfering RNAs. So this is the same as before. But then when we compare important genes to less important genes, we can see that less important genes are much more highly targeted by small RNAs, especially when they have been captured by a transposable element. So this shows that gene capture leads to higher small RNA mapping, and this is even stronger for genes that are less important. Then looking at DNA methylation, here in the 3 contexts, CG, CHG, and CHH. In the top panel, you have synthelogs, which are important genes. On the bottom panel, translocated genes, which are less important. And what we can see here on this graph is that important genes maintain low levels of methylation. So they are all between 0 and 20, 30% methylation level. And this is quite different compared to less important genes, which are quite highly methylated, up to 100% of methylation, cytosines methylated. And donor genes that have been captured are even more methylated than free genes. So this shows again that gene capture leads to DNA methylation of the gene that was captured, even more when it is a less important gene. And we can wonder if this methylation of the donor gene leads to a lower expression of the gene, because then it would impact fitness. So here again, we look at important genes that were captured, donors, and that were not captured, free genes. And here you have their expression level. And you can see that the genes that were captured, but that are important, maintain a very high expression level. So it shows that for important genes, the capture doesn't affect expression level. And this is quite different for less important genes, because when these genes were captured by a transposable element, their expression level becomes really very low, almost non-expressed genes. So we can confirm our hypothesis that selection limits the trans-epigenetic effects of gene capture for important genes that we identified as synthelogs here in our dataset. So next, we wanted to see whether the repressive small RNA mapping and DNA methylation remains in the captured region or whether it spreads beyond this captured region. So here in this graph, you have the captured region in light green and the non-captured region in dark green. And here you have the small RNA mapping per kb in the captured region. And you can see it is much higher than the non-captured region for important genes, so synthelogs. So for important genes, the small RNA mapping is restricted to the captured region and it doesn't go beyond. And the observation is quite different on less important genes, so translocated ones. because they have similar small RNA mapping between the captured and the non-captured region. This is not significantly different, which shows that for less important genes, small RNA map on the entire length of the gene. And now looking at DNA methylation, so in the three contexts, here on the top panel, you have important genes, synthelogs. And what we see is that the captured region in light green is highly methylated in these important genes. However, the non-captured region in dark green remains lowly methylated. So for important genes, only the captured fragment becomes highly methylated and the rest of the gene maintains low methylation level. This is quite different from translocated genes that we call less important, because for these genes, both the captured and the non-captured regions become very highly methylated in the three contexts. So the less important genes become methylated in their entire length, which then leads them to have lower expression levels as we just saw. So we could confirm our three first hypotheses. So then on to the last one, which is that maybe the transposable element benefits from the capture, which I illustrated here as a wolf in sheep clothing. So the parasite hiding from the host. So the first thing to look whether there is an advantage for transposable element is to look at transposable element age. So for this, we computed the terminal branch length of the transposable element, and this is an estimate for the age. And we compared Ts that captured as an important gene, a synthelog, compared to Ts that did not capture a gene. And what we see is that the transposable elements that capture the gene tend to, they are significantly older than free transposable elements, which suggests that after gene capture, a transposable element is able to remain for a longer time in the genome. So maybe it's advantageous to capture a gene. Here we look at the level of small RNA mapping per kb on the transposable element, depending on whether it captured a gene here in red, and or if it's free in light blue. And you can see that free transposable elements map significantly more small RNA compared to transposable elements that did capture a gene, which again suggests that it's advantageous for the transposable element to capture a gene, because then it is targeted by fewer small interfering RNAs. But when we looked at methylation level, here in the three different contexts, we found that transposable elements are very highly methylated, close to 100% of cytosines methylated in the CG context. And there is a small difference between the Ts that captured and the Ts that did not capture a gene. But at this level of methylation, the T is silenced anyway. So we conclude that there is no advantage for the transposable element to capture a gene, because in any case, it becomes silenced because it's very highly methylated. So we conclude that we cannot see a strong advantage for gene capture for the transposable element. So to conclude on this second part, we found that gene capture, when it happens with an important gene that we inferred as synthelogs here, then there is indeed small RNA that can map both on the donor gene and on the transposable element. So there is a small RNA crosstalk. but these small RNA do not spread beyond the captured sequence of the gene. And similarly, for the DNA methylation, it doesn't spread beyond the captured sequence. And this maintains expression level of important genes. The story is quite different for genes that are less important, which we identified here as translocated genes, because these genes, after gene capture, they become very highly methylated, and they become silenced. So they lose most of their expression. With that, I'm done, and I would like to thank all the people who contributed to these two projects that I presented today. And thanks to the organizers of this seminar for inviting me. And thank you all for your attention, and I'll be happy to take questions.
Thank you very much. It raises a lot of questions, but I think I won't have the time to ask any, also because there are already plenty of questions coming from the audience. So I think the first one was Nicola Roder. So Nicola, if you want, then you can speak aloud.
Yeah, thank you, Aline, for this very interesting talk. So my question was relative to the first part. I was wondering if genes that are gene body methylated, if they tend to cluster together in the genome, and if this occurs, how does your test account for this? So for the fact that, for example, some region in the genome, because of the chromatin context, the genes in these regions, they will all be gene body methylated, and so that the distribution of genes along the genome could affect the results of your test?
Yes, that's a good question. I haven't controlled in my analysis for the location of the genes, and I'm actually not sure if there is a bias for that. In Arabidopsis thaliana, I don't think there is a clear clustering, and I think the gene body methylated genes are located everywhere in the genome. But I could check. To be honest, I'm not sure.
Yeah, so I'm thinking about having a model where you would take the location of the gene into account with some kind of spatial autocorrelation, and then you would account. So if two genes are gene body methylated and they are close to each other, you would account with this in your model and adjust the test accordingly.
Okay.
I don't know. If they're randomly distributed, then there is no issue, of course.
Okay. Thank you.
Thank you.
Thank you very much. Then we have a question by Frank Johannes. So Frank, if you want, then you can please ask your question aloud.
Hello?
Yep.
Oh, it's my turn. Okay. Hello. Let me see what my question was. Yeah, so this is about the first part about GBM variation. There's always some challenging interpretations of evidence of selection, because GBM variation could be the result of cis and transacting genetic variants, right? And
yes,
they would lead to a departure from the neutral methylation side frequency spectrum and mimic selection. So how do you deal with this alternative explanation?
Yeah, so of course, it's always very hard to disentangle genetics and epigenetics. And in our SFS, we don't separate them. So we don't correct for these trans and cis effects. In the expression analysis, we looked at this effect when we looked at the association between higher expression and gene body methylation. We put in the model the amount of CG sites, and we saw that the effect of the amount of CG sites was going in the opposite direction. So it wasn't contributing to the effect of GBM on expression level. It's always a very hard.
Yeah, I know
it's always very hard. I think I quite liked the way that Daniel Zibelman looked at it in his recent preprint. So the way he did it was to find important variants with GWAS, and then he did a subsampling of the accessions that had the same variant.
Yes.
So I think that's clever, and I think it controls for the genetic effects quite well.
Right.
In the SFS, so we don't control for genetics, but because epimutation epimutates more quickly, well, of course it could be a problem, but I think it's also fine. But it's discussed. It can be discussed, of course.
Right. I mean, I think the conclusion is a bit strong that there's selection of GBM. Yeah, if there are alternative explanations, and I mean, what Zilberman is doing, yeah, this is the way to go, I think. But a slightly different way to do it is maybe to rely on methylation QTL from that have been mapped already in this population, and then work with the residuals for GBM. And see if you can still get, you know, any evidence for selection of GBM. You know what I mean?
Yes, yes. Are you going to do it?
Well, no, maybe, I don't know.
Okay. Well, thanks for joining from very far from Montpellier.
Okay, well, thanks for the talk.
Thank you.
Thank you very much. We have many questions open. We have one question by Aline Muyle. I'm not sure I have, so Aline, if you want, then please to speak about your question.
Well, yeah, we were sort of curious of like if some plastic traits are mediated by epigenetic change, maybe from one trait to another. For example, phenology is one of those traits highly plastic, depending on environmental fluctuations. And it was also one of the traits that was like highly methylated. How could selection account for this? For example, if it's a highly fluctuating environment, or for example, if these methylation changes are not kept in a phylogenetic scale as was presented in the first part. Is there a possible way to see if there is any selection going on for this?
I'm not sure I get the question. I'm sorry. Could you be more specific?
Okay, yeah. So for example, if there is adaptive plastic responses to environmental cues that are driven by epigenetic changes, could there be any way to measure selection for that?
So I think it's quite complex because again, there is this coupling of genetic and epigenetic that we speak a lot about today. And so a plastic response mediated by epigenetics could be under genetic control. And I guess the first thing would be to check what is the epigenetic mark causing this effect. Because if it's CHG, CHH, it's mostly genetic effect. Then if it is CG methylation, then it can be inherited. So it could be under selection, theoretically. Then to test if it's under selection, you can use different approaches. There has been actually a method derived from population genetics. It's I could I don't have the reference in mind right now. Yeah, maybe I can send you something by email. Sorry. There are different methods. So the SFS looking at just one locus, it doesn't work. So then you can do QTL like we were talking about previously. There are very different ways. The difficult thing is always to disentangle the genetic and the epigenetic effects.
Yeah, okay, well, thank you very much. And yeah, if you could share any reference, that would be
Yes, sorry, I don't have the reference in mind right now.
Yeah, great, thank you.
Yeah, thanks. I think we have for them probably a question for Yanis Michalakis, you asked, so maybe you can pick and choose one question among all the three that you proposed, please.
But Patrice is younger than I am.
Yanis, go ahead.
I was afraid of breaking the rules. Thank you for this very interesting talk. Well, I guess I'll start with my first question, which is simply whether gene capture may explain the results of the first part, that is whether ancestrally methylated genes tend to be more gene captured than ancestrally unmethylated genes.
I haven't looked into that particular question. So it could be. I mean, it's very difficult to test because transposable element turnover is quite fast, and so we don't know. We can only see recent capture events, and I haven't compared gene body methylated and unmethylated genes, but yeah, I don't think there was an effect. But I see where you're going for. So it could be that gene capture leads to methylation of the gene, and then only CG methylation remains with time.
Yes.
Once the capture event is old, and then this CG methylation leads to gene body methylated genes. So this could be a hypothesis. We could also have a similar effect in cis when a transposable element is nearby a gene and the methylation spreads and the gene becomes methylated in the three contexts. And then over time, the transposable element is lost and only CG methylation remains. So yeah, I think it could be a driving force for gene body methylation evolution.
Okay, thank you. I may come back again later if the chairman allows it.
We have one question very rapidly, please, by Iago Bonici. Iago?
Here, can you hear me?
Yep.
Yeah, I was just naively wondering, under the conflict model, the transposable element capturing important genes lead to these genes being more methylated, which in turn affects their expression level, which if I understand correctly. And I was wondering whether this could be a natural way to make this this epigenetic selection adaptation run. Like, would it be a natural way to silence genes for short periods of time across evolution, or would it be completely anecdotic?
Aline, your microphone is muted.
Sorry, I was drinking and then I forgot. So yeah, that's the exact hypothesis that we made. Sorry if I didn't make it clear in my presentation.
Yeah,
so
it was the fourth one, right? Yeah.
So we think that when a gene gets captured, it often gets moved at the same time. We cannot prove it, but it's we can see this association. And then we think that this is leading the gene to pseudogenization, because then it lands into a bad neighborhood with heterochromatin, and it becomes less expressed and just degenerates.
Yeah, great. So it's the fourth one that you haven't been able to prove, but you actually think it could be a way, right?
Yes.
Okay.
Yes, we cannot prove it right now. It's quite difficult to prove, and we would need looking at different species, more species than we have. Well, it's complicated. It's doable, but complicated.
I understand. Yeah, thank you.
Just need to plug my battery.
Probably we can have a last question, and then probably a very brief answer done by Patrice David. If you want them, please.
Okay, do you hear me?
Yep.
Yes.
Yeah, that was a question. Maybe it's something I just didn't really understand, and I'm quite inexperienced in the domain, but it was related to the first part of the talk when you look at the frequency spectra of the methylations, gene body methylations, and you compared the spectra of genes that are ancestrally methylated and genes that were not ancestrally methylated, and you concluded from that that there was probably some selection going on to distort the frequency spectra and make them different. And so it's based on the Charles and Jane model adapted to increase the mutation rate. And I was wondering that even if we consider that the mutation rates are very high, so the pace of trajectory to equilibrium can be quite rapid, but still, is it not just possible that the frequency spectra keep the traces of the ancestral state, and that's why they look so different?
So
that we will not be at equilibrium. So the site frequency spectra are just, you know, still similar to their ancestral state because they haven't got to.
So I think we did a control that answers that question. I think that I haven't shown. So we did different groups of genes that we know have different epimutation rates. And when we consider these genes separately, because some of them epimutates more highly, some of them more slowly, and we can still observe the same conclusions that the genes that are ancestrally gene body methylated are under selection. I think it's not impacting our results, but I'm not sure how to demonstrate it other than by this control that we did.
So the fast epimutation genes have the same frequency distribution as the slow ones?
They have a slightly different SFS, but the conclusion is the same.
Okay.
I'm not sure it helps, but.
Yeah, okay. Yeah, I think it does.
Thank you.
Okay, thank you very much. I think I'm afraid we need to stop here. We are over the time. Thank you very much once again, Aline, for this talk. That stimulates a lot of thought. And then so for next week, I would like to remind you that we will meet at 16 p.m. So at 4 p.m. So the speaker is based in Princeton. So Jonathan Levin. And then with this, so thanks again for all this attendance. It has been a very good one. Thanks again, Aline, for your talk. And then enjoy the weekend. Bye.
Thank you all for coming. Bye.